It's been a rocky start to the semester for me. At least this time, it's not that I'm missing a lecture, it's that I didn't have the notes posted. Okay, so yeah, if you checked before and you didn't find this on campus, it is there now, because Heather put it up there. Okay, you ready? Ready to rumble? Went through, I watched way too much football this weekend, including the Browns game. They tie, and they keep saying things like, a tie is like kissing your sister, and I keep thinking, what's odd about that? So, what we're going to do today is work on signaling systems. And I know this because there's this pretty slide up here that says signaling systems. And I do forewarn you, don't get used to it. This is the only fancy slide that I have. And I don't remember at this point, have I only given you fancy slides so far? Yeah. Okay. Well, this is the way my slides usually work. I feel a lot more comfortable around them. That's basically the way I lay out slides is write it in hand, and as though I was taking notes. Oh, no. Okay? I print very well.
You do have beautiful
Okay?
handwriting.
For the most part, things are pretty clear. So this is all about me and my comfort level, and maybe you start to feel more comfortable with it as you go along. So, once we became multicellular organisms, we started to specialize. So, if you're a cell and you're a prokaryote, it's like having carry-on luggage. You got to fit everything you need into that one cell, and then you go about your business, and you count on yourself for survival. But if you become multicellular, eukaryotic cells, what starts to happen is you lose some of the ability to survive on your own. You develop special abilities to help the group as a whole to survive. But as we start to do that and differentiate into different cell types, what happens is we need to rely on other cells for our own survival. Sort of like PBL, solving PBL. Some people don't like that. Some people want to do it all. So, and some people don't want to do any of it. Those are the cells that die. So, your future, and I'm laughing, right? So, as we specialize, we need to depend on other cells, and therefore, we need to be able to communicate. And so we develop this ability to signal between cells, to coordinate our efforts, to be able to do whatever needs to be done. And together, if we all do our own job, but we do it in a coordinated manner, then we survive. So signaling systems became more and more important as cells became more and more specialized. And we can kind of get an idea about that from the vast array of different signaling systems that we see in cells. And what I'm going to talk about today is just kind of mostly in general terms, but I'll give you a few specific examples of different signaling systems. This week, you'll probably get into more detail on signaling systems as well as we go through our PBL. So what we're going to look at, and I think I started off early on, there are three in the course. There are three jobs every cell has to do. The first job is to survive, because if it doesn't survive, it doesn't have to do anything else. But then the second job is to sense, specifically to sense its surroundings and know what's going on. And then the third job is to respond. It does you absolutely no good to sense your surroundings if you don't change your behavior, if you don't respond to whatever you're sensing is going on. So surviving, we're going to talk about pretty soon. We go through how do we make enough ATP to make sure we can continually combat the universe. But here we're going to look at how do we sense our surroundings, and then transition that into how do we generate responses. So signaling systems is about sensing and responding, and mostly that's involving communication between cells. However, we do have some cells that are specialized enough to sense our external environment. So we'll talk about in the spring glucose-sensitive neurons as part in the brain that is going to cause you to feel hungry or to feel satiated. We have retinal cells, rods and cones that can transduce light energy and then through a sequence of communications produce frequencies of action potentials in ganglion cells that we perceive as vision. So we've got specialized too. But here with signaling systems, I'm thinking primarily communicating between cells. If I start thinking about that, I have to think about the response that I'm going to get, both in terms of being able to sense something and then to generate a response has to involve proteins, because proteins do active functions in the cell. Okay? So any response by a cell is simply going to be a change in the total activity of a protein or group of proteins. So the first thing I have to define here is when I'm looking for a response, I've now got it. What's a response? It's a change in the function of a protein, total activity of a protein. So the next question I ask is, what do I mean by total activity? And total activity of a protein is simply equal to the specific activity of each individual protein molecule times how many of those molecules you have. So the amount of that protein. So if, for instance, so let's take an enzyme that we already all know, and that's hexokinase from last week. So if we were talking about the total activity of hexokinase in a cell, a key regulatory enzyme for glycolysis, what would it be equal to? It would be equal to how quickly each individual molecule of hexokinase can convert glucose to glucose 6-phosphate. That's specific activity. How fast each individual molecule can do that. And then we multiply that times the total number of molecules of hexokinase that we find in that cell. That's the total amount of the hexokinase. Okay, so what signaling systems have to do is change the total activity of an enzyme. And I can see from that equation up above there, we have two ways that we could do it. We could either increase or decrease the specific activity of each enzyme molecule, or we could change the total amount of the enzyme. The example that keeps coming to my mind year after year, and I don't know why, is let's say the dean came down and he said, Tim, I've got a bunch of letters I need to send out to alumni asking them for donations. You'll find out about this even before you graduate. The day you get your diploma at the commencement, at the graduation or diploma exercise, you'll have a talk by somebody who is going to tell you, we're going to be sending you a letter. So they're communicating with you right then. So let's say the dean is sending out some of those letters. He said, I got 200 of these letters and 200 envelopes, and we need to send these out right away. So Tim, what I need you to do is fold those letters, all 200 of them, and stick them in the envelopes for me. Can you do that? And I say, sure, you're the dean. Because I'm a fearful guy, you know, basically. But what am I going to do? Because I've got to sit here and talk to you. You're expecting that for your $71,000 in tuition. So what I'm going to do is go to, what's your name?
Sam.
Sam. You sure? Okay. Got to go to Sam and go say, Sam, here's 200 envelopes and 200 letters. What I need you to do while I'm talking here, don't worry, Her Highness is taking notes today. You won't have to worry about that. Pulled up those letters, stuff them in the 200 envelopes, and I need it to be done by the end of class. Sam says, sure, because he's fearful too. And I give him his final exam in FLS. Leverage, that's what it's called. So Sam starts folding, starts stuffing. I keep talking. And while I'm talking, you know I'm easily distracted now. I keep talking, I look over, and Sam is not going to finish up this job. He's not going to get all 200 letters folded and stuffed into the 200 envelopes by the end of class. So I have two choices because I don't want to disappoint the dean. Okay? My two choices are, one, I could get Sam to work faster. That's okay. I came prepared. I'm from Wisconsin, America's dairyland. We all own cattle prods. These are little sticks that give electrical shocks. So I go back behind, now look at the fear in Sam's eyes, go back behind the podium, get my cattle prod, and zap Sam. You know what? It gets Sam's attention. He starts folding faster. Why? Because he doesn't want to get zapped again, right? Oh, by the way, my daughter is a social worker at UH. She had a patient or a client, I guess they called them, that was in the hospital and was getting released. She'd gotten a concussion, and the reason she got a concussion, I have no idea who the patient's name is, so I can share this information. Because her dog had one of those shock collars for training them, and she wondered how uncomfortable it was for the dog. So she put the shock collar on and walked beyond the barrier, or outside the barrier, and got a shock, which threw her head back, and she hit the back of her head on the wall and got a concussion. So Sam doesn't do this, don't worry, there's no walls close to Sam. So Sam doesn't want another shock. Neither did that woman in the hospital. So he starts working faster, but he is human. I think you can picture that, oh boy, I don't want to get that shock, I don't want to get that shock. And as I continue talking and evidently ignoring him, he just kind of naturally goes back to his intrinsic rate of letter folding and stuffing. And so over time, the effect of the shock wore off. That's okay. My cattle prod comes with multiple shocks in it. So I can shock him again, he'll start working again faster, but it wears away pretty quickly. So that's one way. That was specific activity. I could increase the total activity by altering Sam's specific activity. Or I could go and I could say instead, Jake. Sam and Jake. We got these good wholesome names up front. Things I can remember. Okay? Like Tim. That's not wholesome. Okay. So I could go to Jake and I say, here, Sam, I don't want to give you any shocks anymore. I'm going to give half of the undone ones to Jake. So now I got two of them working. And the job is getting closer and closer to being done. I still got my cattle prod. I could give both of them shocks if I wanted to. But the more people I incorporate, the more total activity I have. So that is the example that always comes to mind. So we can alter specific activity or we can alter total amount. Okay? If we want to alter total amount, if we're talking about proteins, what we have to do is make more proteins or degrade some. Let's picture, it's easier to picture making some. So if I want to increase total amount to increase total activity, what I have to do is go into the nucleus, send a signal into the nucleus saying we need to transcribe this gene, process it, make some messenger RNA, take the messenger RNA, send it out to the cytosol, have ribosomes assemble on it, translate the message in the messenger RNA to make an initial new protein. Then I have to do some post-translational processing for most proteins, altering it here or there until I have a fully functional Jake. Okay? So it's a little bit like Sam has to reproduce to produce Jake. That's going to take time. There's a reason for these really weird analogies because they tend to stick in your mind. I know this because students report waking up in the middle of the night screaming in terror because they were dreaming about this stuff. So it takes time to make more. It took no time at all to give Jake a shock, or to give Sam a shock. It would take no time at all to give Jake a shock either.
People get there very quickly.
So altering specific activity, this is going to be fast onset. We just need to do something to the molecule. This is going to take us a while to make. So we'll come back to that as we go through different types of signaling. Yeah?
For specific activity, is it per molecule of protein or is it per active site of the protein? If they have more
It's
than one active
per
site.
molecule of the protein, yeah. The active site generally, you generally just catalyze in one specific reaction. There aren't too many, it's no protein I can think of off the top of my head that is doing, catalyzes two different
If it's like a
reactions.
dimer or like a Kitchener with an active site.
Yeah, but it's still, once it's like tyrosine kinase, once it dimerizes and becomes activated, then it's still just doing one reaction at a time. Okay, so the word signaling, and by the way, there's only one L in signaling. I've checked it several times. It keeps coming up as just one L. I am convinced, though, there should be two. In Tim's world, there are two. So when I write things out by hand, there's two L's in signaling. Spell check usually catches that if I'm typing it. So the term signaling implies something. It implies to me that information is being transferred, that's being communicated from one place to another. So a cell or one part of the cell knows something, and now we need to let some other part of the cell or some other cell know what's going on. But the important part is that it's information that's being transferred. So that brought to mind the next question, which is, what are some examples of signals in our body and signaling systems? So I started dividing it into groups, signals into groups. We're going to start with the fastest. The fastest type of signaling we can find in our body is for direct electrical communication. The flow of a membrane potential change from one cell to another. So we've talked about action potentials. We know how to do the Macarena. We can generate action potential. Now, once I've generated an action potential somewhere, how do I get it to go somewhere else? Well, we'll talk about how we get, next year, we'll talk about how we can get an action potential to move. It's a pretty simple process. But what if we wanted the information represented by that action potential to be transferred to an entirely different cell? Not just to move along the axon of a neuron, but to move from one cell to another. And that would be direct electrical communication. That would be if we generated an action potential in cell A, that it moved into cell B. And the way we allow that to happen is to have gap junctions, kind of a fusion at one point of two cell membranes. And so we have the ability, we have channels of cytosol that allow the direct flow of the action potential from cell A to cell B. There's hardly any delay. Okay, this action potential can move rapidly along the membrane. The gap junction represents a higher resistance pathway than just the flow along the membrane. So it may be a little bit slowed down there, but it pops through these gap junctions very quickly and moves on to cell B. Yeah,
Can one cell potentially be connected to many others?
yes.
So so there's no real directionality, this is just a diffuse.
Yeah, I mean, if we excited cell B, it could go up to A.
or
We
CD.
could put a bunch of cells all around here. But the consequence of that, we'll come back to, the consequence is, if one cell has an action potential, all of the other cells that are connected are going to have action potentials as well. And I can't think of gap junctions and direct current flow without thinking about the heart. The heart is the poster child for having gap junctions, direct electrical flow. Current flows from one ventricular cell to the next, one atrial cell to the next. And so if we just think, limit right now, say ventricle, if one ventricular cell depolarizes, they're all going to depolarize in short order. Okay, we count on that. Because what happens, how many of you have had physiology? Okay, this is going to be a really deep question. What happens when a ventricular cell depolarizes? What's the purpose in life of a ventricular cell?
Contraction.
To
Right.
contract. What causes it to contract? It has an action potential. So if a ventricular cell generates an action potential, it's going to contract. Now, if we get one ventricular cell, just one ventricular cell to contract, what kind of effect is that going to have on the blood?
You know,
Nothing. Okay, one ventricular cell contracting is not going to generate enough tension to get blood to move out of the ventricle, if we're talking the left ventricle, into the aorta. No, but if we get them all together, and we get them all contracting at once, the ventricular walls press in, ventricular pressure goes up, exceeds 80 millimeters mercury, and ejects blood into the aorta. If we all work together. Okay? So we count on this in the heart. We count on rapidly, if one ventricular cell depolarizes, we want them all to depolarize in short order because they're all then going to contract together. So that's the strength of electrical flow. Electrotonic conduction is a fancy word for that. Okay? But it's got a problem too. Problem is, going right back to what we're counting on, that if we get one cell to depolarize, they're all going to depolarize. If one cell has an action potential, all of you will have action potentials. Okay? Yeah.
Okay, so, but there is directionality in those Purkinje fibers, just not directionality in the heart cell.
There's directionality, yes, built in, especially I think of it more for the AV node.
That's a-
And I can't,
But you get
I
down
can't
there,
figure
and
out
then
how
it's-
that works.
It's diffuse.
Yeah.
At the bottom, but not the top.
yeah, if you stimulate one part of the ventricle, just get a stimulus, you'll, seen enough medical shows that, the patient is flatlining or something, and so they have cracked open the patient's chest, there's the heart, and they put those two little paddles directly on, and they stimulate it, and the heart suddenly contracts. Well, that was two different points where they normally electrical stimulation isn't occurring in the heart, but it flows all the way through the ventricle. So it's multi-directional. Okay? Well, if it's so fast, and easily the fastest type of communication we have, if it's so fast, how come we didn't program our computers to work that way? Our brain. Brains are amazing how much they can do, and they all work on those ones and zeros, right? Generating action potentials, and this frequency of action potentials in this neuron means something. That's how information's encoded. And then they communicate with other neurons. And that's primarily through chemical signaling, neurotransmitters. Well, why did we, if we had a faster option, why did we just go with the slower means of communication? Next year, we're going to talk about we can speed up conduction of action potentials by making larger diameter axons or by myelinating them. Speed, speed, speed. But then when it comes to communicating between neurons, we go with the slow option. Why is that when we've got electrotonic conduction?
Long-term effect.
Well, because the same thing that makes electrotonic conduction, direct current flow, very valuable to us, makes it not valuable to the brain. Because what that would be, if we used electrotonic conduction throughout the brain, if one neuron had an action potential, they all would have an action potential. When do we see our brains kind of operating like that?
Seizure.
In a seizure, where they're doing it quickly, or in stage 4 sleep, or stage 3 and 4 sleep, deep sleep, or / coma, where you see these big waves of simultaneous electrical activity. Seizures aren't a good way for your brain to process information. Okay? What makes your brain powerful is the ability of each and every neuron to make up its own mind, to decide whether or not it should fire. And so how electrotonic conduction is rapid, there's no flexibility. One cell's going to behave like all the others. And that behavior, if we all behave like the same cell, that's called a functional syncytial. That functionally, we're all behaving like one big network behaving as one. So, anybody know, is the heart the only place we find electrical conduction? The answer is no. Where else in our body do we use electrical conduction?
Would it be anywhere there's that junction?
Yep, so where would you find them?
Everywhere. Everywhere.
Yeah,
No,
anywhere-
not
Anywhere cells are held together.
excitable, no. We got tight junctions. Smooth muscle, but not all smooth muscles. So you're the assistant, it turns up the volume. It's good. Smooth muscle, what type of smooth muscle? Anybody happen to remember?
Peristaltic?
Peristaltic, yes. And that we want one ring after another to contract. And so that would be an example of single unit smooth muscle. There's single unit and multi-unit. You'll learn that exactly 1 year from today. The second years are studying for their muscle physiology test now. Okay, and also there is evidence for some electrical conduction in the brain, that we have direct ties, that one neuron fires, the other one will too. And for a while they thought this might be the cause of generating seizures, but not good supporting evidence for that. Okay, so let's move to chemical signals. There are, the moment we say chemical signals, they're slower than electrical. Okay? But they're a lot more flexible, and they're also a lot more diverse because we can use a lot of different chemicals as signals. So we're going to divide them here into 3 main types. And the first one is going to be neurotransmitters. Neurotransmitters are the fastest of the chemical signals. Okay? They produce fast onset, short duration responses. Presynaptic terminal exocytosis, a neurotransmitter for the most part. This is the way it works. It binds, it traverses a very small distance called the synaptic cleft before it can bump into a receptor. It binds to the receptor, which generates some change in the postsynaptic cell's behavior. We'll get into those specifics in a little bit. Okay? What makes chemical signaling for neurotransmitters so fast is the synaptic cleft is small. So there's very little diffusional distance that has to happen in order to bump into a receptor. But fast relative to other chemical signaling and short duration responses. Okay, we'll see again next year, we generate things called EPSPs or IPSPs, excitatory or inhibitory postsynaptic potential. That lasts for maybe 5 milliseconds, maybe up to 20 milliseconds. The second type of chemical signals would be the hormones. Now hormones go to the other end of the chemical signaling spectrum. We're going to divide hormones, so they tend to be slower, definitely slower than neurotransmitters. So hormones in general, slower onset responses, but longer duration responses than what we would see for a neurotransmitter. But if we're going to talk about hormones, we need to divide them into two major subtypes or subcategories of hormones. There are the polypeptides, or polypeptide-like in function, and the steroids. Okay? What you need to remember right now, and we'll talk about this again next year when we talk about endocrinology, we'll remind ourselves, polypeptide hormones, they're like short proteins, right? Amino acid polymers. They're working by altering specific activity, primarily. So they're going to act like cattle prods. And they're going to be, of the hormones, they're the faster onset group and shorter duration response. So when you think of polypeptide hormone, think of Sam, and think of me treating Sam with a cattle prod. Okay? And so when I see polypeptide hormone, what immediately pops into my mind, fast onset, short acting. If, on the other hand, we're dealing with a steroid hormone, okay, what they do is work by altering gene expression. If we do, again, primarily, we have cross-signaling going on in certain cases. But if we're altering gene expression, it's going to take a while to make a new protein. So when I see steroid hormone, I think slow onset, but long duration response, because once I make this hormone molecule, once I make this protein, it's going to be around forever, or not forever, for a long time. So when you think steroid hormone, I want you to remember Jake, okay? Because Jake had to be reproduced, gestated, and then be born. But once he was born, he's going to be around working for a long time, right?
I don't appreciate it. I'm natty.
Yeah, you are. Okay?
All hard work.
All hard work and no play. Okay. So, polypeptide steroids. Fast onset, short acting, slow onset, long acting. And then the third type of chemical signal would be faster than the hormones, because the hormones circulate in the blood. So it takes them quite a while to get to wherever they're going. When they hit their target cell, well, they had to get there by moving through the blood. How did they know it was a target cell? Because it had a receptor for them. Now the local signals are going to be faster than the hormones, but tend to be slower than neurotransmitters. These local signals are characterized by short lifespans. Okay? They don't have time to circulate in the blood. They'll be catabolized before they got anywhere. So short lifespans, can't circulate in the blood, so they're not endocrines. Instead, they work nearby, wherever they're produced, and we have two major subclasses of them. They're not endocrines, but they could be paracrines, which means para, nearby, or next to, or autocrines, which means the same cell that produced them responds to their presence. So paracrines are released by one cell and they affect neighboring cells. And the onset and duration varies, but onset is going to be short, a little longer than a neurotransmitter. Duration oftentimes will be significantly longer than a neurotransmitter. Okay? Some examples would be, the first one you to think of would be prostaglandins, which are, the whole icosanoid group, prostaglandins, thromboxanes, leukotrienes, are all paracrines. Histamine, the kinins, all of those are examples of paracrines. And then autocrines. Autocrine is produced by a cell, released by a cell, and it turns out that the cell that produced it and released it actually has a receptor for it on the very cell membrane that produced it. And so it starts a cell's behavior, and the signal it's sending out starts to affect the cell that was producing it, which seems kind of weird, but it has different effects or different values. So an example, the one that I keep in my mind of an autocrine is adenosine. Okay? So what's adenosine? Anybody know? What's ATP?
Adenosine triphosphate.
Adenosine triphosphate.
There you go.
So, what's adenosine? It's ATP without its TP. Doesn't have any phosphate. And so I know ATP is a purine nucleotide, which means it has a purine base, adenine, bound to a ribose with at least one phosphate bound to that. So ATP would be an adenine bound to the carbon number 1 of ribose, and then 3 phosphates linked end to end on carbon number 5 of ribose. That's ATP. And that's a nucleotide. Adenosine would happen if we clipped off the 3 phosphates. So it's no longer a nucleotide, it is a nucleoside. It's just the adenine bound to carbon number 1 of a ribose. Okay? And you can make adenine by just having the cells successively dephosphorylating ATP to ADP to AMP and finally to adenosine. Okay. Now, how do we see this adenosine working as an autocrine? The picture I have is for a neurotransmitter. A neurotransmitter, let's say acetylcholine at a neuromuscular junction. And we load up a vesicle with acetylcholine. And how do we load up that vesicle with acetylcholine? How many of you knew that we load up acetylcholine molecules in vesicles for some neurotransmission? Okay? How did that happen? How did that come to be? Did all those acetylcholines say, I want to be a part of the group too? Or do you think they got dragged kicking and screaming into that vesicle?
Yes, especially against your